Good morning, students. Welcome to Biodimension online class. And in today's class, we are going to discuss the topic, and that is fates of pyruvate and fermentation. So let's start our today's class. So in today's class, we are going to discuss first the fates of pyruvate. So pyruvate, what is pyruvate? Pyruvate is the end product of glycolysis. So pyruvate have some importance because depending on the condition of body, the pyruvate can go under different kinds of reactions. So here we are talking about all these things. So now, as we already know, glucose after the process of glycolysis, which have 10 reaction steps, it converts into two molecules of pyruvate. Glucose is 6 carbon sugar, pyruvate is 3 carbon sugar. So in this particular process, 6 carbon sugar, 1 6 carbon sugar is converted into 2 3 carbon sugars. Okay. So now here is the main condition based on which pyruvate will decide its fate, and the basic conditions are presence of oxygen. So we can say that pyruvate have different fates based on either they face a aerobic condition or an anaerobic condition. If they face aerobic condition, they will directly go for the production of acetyl CoA, followed by the complete degradation of that acetyl CoA to form carbon dioxide and water through citric acid cycle. And in this pathway, they are actually able to produce NADH and FADH, NADH and FADH2, as well as GTP. So these are energy sources that is being produced via the aerobic pathways. So these high energy compounds will subsequently converted into ATP via the process of electron transport chain and oxidative phosphorylation. But if that pyruvate encounters anaerobic condition, that anaerobic condition may generate in case of the bacteria or the prokaryotic organism, or in case of the eukaryotic organism. So in our body also, this anaerobic condition may get generated. So in case of hypoxic or anaerobic condition, the pyruvate get fermented into ethanol. In case of eukaryotic cells, pyruvate get converted into lactate. So here in this particular class, we are going to discuss both of this process of fermentation. At the very end, I will show you that these two things are not the end of this fermentation process. Based on the type of bacteria or type of organism, different kinds of fermented product may get generated. That is not the purview of this class, but yes, the fermentation is quite a large topic, and if you want, I will definitely make a video regarding fermentation and what different kinds of fermentations are. And we need to know one thing, that fermentation, whatever it is an industrially important process. But here, our point of view is not based on industrial utilization or industrial importance of fermentation, but we are going to discuss actually the biochemical pathway which is actually there to complete this fermentation process. So the first thing is that pyruvate may get converted to lactate. The pyruvate may get converted to acetyl CoA via further oxidation. It converted to complete energy, that is ATP. It generates complete energy, that is ATP. So it is in this process of pyruvate to acetyl CoA conversion, when one carbon dioxide is being removed or released from that system. So, and the third one is pyruvate to ethanol production. In pyruvate to ethanol production, this is also a two-step process. Pyruvate to acetaldehyde. Here, acetaldehyde is a 2 carbon compound, and in this process, pyruvate is a 3 carbon compound. So one carbon dioxide is being released. So the first step is decarboxylation step. So this is a decarboxylation. In the second step, acetaldehyde is being converted to ethanol. And if you are looking at, if you look at these two particular technique, in these two particular technique, what is common? Just try to answer that in these two particular technique, what is common? The common is the conversion of NADH to NAD plus. So this conversion of NADH to NAD plus is important because as it is an anaerobic process, and in anaerobic process, NADH need to get converted to NAD plus, or else the scarcity of NAD plus will arise, and if NAD plus scarcity will arise, that will hamper the process of glycolysis. So this is a process. Fermentation is a process by which NAD plus is getting recycled, or NAD plus is getting oxidized, or in the other hand, pyruvate is getting reduced. So NAD plus is getting oxidized, and that is a very important part of this particular step of fermentation. So as I have already told you that rates of pyruvate in aerobic condition, the NADH formed in glycolysis must be recycled to generate NAD plus, and that is why several kinds of electron transport mediums are there, and through that electron transport medium, the NAD plus is getting converted to NAD. Sorry, NADH is getting converted to NAD plus, and the oxygen is actually acting as a terminal electron acceptor for the completion of this respiration process. Now, if we look at that, whether it's aerobic one or it's anaerobic one, the main concept of conversion of pyruvate to any other component, one thing is that finally NADH need to get converted to NAD plus. Okay, so only a fraction of energy of glucose is released in the anaerobic conversion, but in case of aerobic conversion, complete energy is being released via electron transport chain oxidative phosphorylation. I will definitely make a video in detail about electron transport chain and oxidative phosphorylation, but in between this electron transport chain and oxidative phosphorylation, there are several things are there which you need to know, and that is why I will definitely prepare more than 2 or 3 videos based on all these particular topic. So now, if we are looking at ethanol fermentation, yes, it's somehow an industrially important technique. Ethanol fermentation requires anaerobic condition. Until and unless it's an anaerobic condition, it will not work in a proper way. So anaerobic condition means absence of oxygen or presence of carbon dioxide or whatever the other gases except oxygen. So in this particular case, the pyruvate is being first converted to acetaldehyde. As I have already told you, pyruvate is a 3-carbon compound, acetaldehyde is a 2-carbon compound. So one decarboxylation step is here, and as the step is decarboxylation, the enzyme which is involved in this particular step is a decarboxylase enzyme. Now, this in second step, acetaldehyde is converted into ethanol. Acetaldehyde, aldehyde to ethanol. It's a reduction process. Aldehyde to ethanol. If we are looking at the process of oxidation and reduction, acid to aldehyde is a reduction process. Aldehyde to ethanol is a reduction process. But if we go forward simultaneously, alcohol to aldehyde is an oxidation process. Aldehyde to acid formation is an oxidation process. But here what is going on? The thing is that acetaldehyde is converted to ethanol, which is a reduction process. On the other hand, NADH is converted to NAD plus, which is a removal of hydrogen. That is an oxidation process. As we all know, oxidation and reduction is a coupled reaction, right? So the second step is being achieved by using the enzyme alcohol dehydrogenase. So you need to know one thing, that the carbon dioxide which is being released from this particular ethanol fermentation reaction, that carbon dioxide helps to maintain the anaerobic condition also. Okay, so if we are talking about energy calculation of this particular process, yes, glucose is being converted by using 2 phosphate, 2 ADP, and 2 hydrogen. It is converted to 2 ethanol, 2 carbon dioxide, 2 ATP, and 2 water. So if you calculate the total number of carbon, glucose has 6 carbon, ethanol have 4 carbon, 2 ethanol. We are considering 2 ethanol. So if you are not comfortable with 4, you can write 2 into 2 equivalent to 4 plus 2 here. So that is total 6 carbons are there. with the help of production of 2 ATP. Here we are not considering the production of NADH, because the same NADH and NAD plus, whatever NADH is being generated in case of glycolysis, the same NADH is being utilized in case of ethanol fermentation. So somehow it's being neutralized. So we are not considering the production of NADH or NAD plus here in this overall energy calculation process. So now, if we are talk about lactic acid fermentation, unlike the ethanol fermentation, lactic acid fermentation also a very important process in case of several animals. Like, so if we are talking about ourselves, if you just think about that, you suddenly start to go to the gym and suddenly start doing exercises in any means. So what will happen? What will happen? You will face the muscle ache. What is the meaning of muscle ache? Muscle ache means if you start sudden exercise, then what will happen? In your body, oxygen demand will suddenly rise. And to meet that oxygen demand, your body, it is not possible for your body to start supplying oxygen instantly. So what will happen in that case? Your body will generate an anaerobic condition. That means the muscle where the oxygen demand is more, that muscle will generate anaerobic condition. So in that anaerobic condition, whatever pyruvate is being produced, that pyruvate will not able to undergo the process of TCA cycle. That is why that pyruvate will undergo the process of lactic acid fermentation using the enzyme lactate dehydrogenase, and it will produce an product. The name is lactate. Okay, so here also the main motto is to convert the NADH to NAD plus, but it has some other importance also. But before that, if we look at the overall fermentation process, that is exactly same except two lactate. In case of ethanol fermentation, two ethanols are there and two carbon dioxide. But here in case of lactate fermentation, we can able to see that two lactate is being produced. Lactate is a 3 carbon compound, so it's a 3 carbon compound. Glucose is 6 carbon compound, so 2 3 carbon compound is equal to 6. So carbon number matches. So now here also one thing we need to know, that fermentation provides usable energy in absence of oxygen. Yes, in absence of oxygen, complete utilization of pyruvate for the production of energy is not possible. Simply, it's not possible. So, in that case, by going through this fermentation process, our body can generate or can continue the process of glycolysis, and through that process, we can able to generate a little bit or a minimum amount of energy which is required for the minimal activity of our body. Okay, so now I just want to mention one thing here, that lactate which is being used or which is being produced in our muscle, that lactate fermentation also controlled by the pH in our body. That means if lactate productions are there, then suddenly the thing which is happen is that the pH of our muscle will reduced. That means yes, lactic acid is an acid, so lactate is have a acidic part. So our pH of the body will get reduced, and that reduced pH will help to control the overall energy requirement process, and that is why I think we have already discussed it in our previous videos, that is the control of glycolysis. If you have not seen that video, please go to the link which is mentioned above, and please go through that video and learn the process of control of glycolysis. Okay, so now these two fermentation is not the end. Okay, so there are several other kinds of fermentations are there, like glucose to lactate fermentation, lactate to acetate fermentation, glucose to ethanol, ethanol to acetate, arginine to carbon dioxide. So various kinds of fermentations are there, and if you really like to know what are the different kinds of fermentation you want to learn, I will definitely tell you the detail about all those fermentations. So now I will definitely share this presentation with you all, And thank you for watching the whole video. And if you really feel that you like that video and you are able to know several different kinds of things or new informations, please share this video with your friends. And definitely, if you have not subscribed this channel yet, please subscribe this channel and hit the bell icon, so that whenever I am going to publish a video, you are going to get the notification instantly. So see you in our next class. Thank you.